Okay, now I'm going to cover the question number 4 on the practice problems, and this involves silencing starting from the telomere position effect in budding yeast by the Sir complex. So I've just outlined on the board the setup for the problem, and the silencing of the chromosomes in yeast is based on hypoacetylation. So we don't have histone methylation in yeast in the same way that we do in Drosophila and even in Schizosaccharomyces pombe. So in this problem, we have these proteins RAP1, which binds to this anchor at the end of the chromosome, at the telomere, and then we have Sir4, Sir3, which interact with RAP1, Sir2, which is a histone deacetylase, and Sir3 has a special property that it binds to the deacetylated histones that is actually the mark that is going to be spread. So the way that the spreading can occur is that Sir2 deacetylates the histones, and this creates a binding site now for Sir3. Sir3 binds, recruits Sir4 and Sir2, and then that can deacetylate the next set of histones. And so this is how you get this spreading mechanism. So to measure the silencing, we use the reporter gene AD2. And so as you recall, normal yeast have white colonies, and a cell that is deficient for producing AD2 gene product will give colonies that are red, and that's because they build up this it's a biosynthetic pathway, and in AD2 mutant, they are defective in turning over an intermediate, that intermediate has a red pigment, and it causes the cells to be pink. And so when you have the AD2 gene inserted near the end of the telomere, it's going to be subject to the telomere position effect and variegated phenotype, very similar to in the Drosophila eyes. So if you insert the wild type AD2, it should be silenced. And so even though the wild type version of AD2 is present in the chromosome, it's going to be silenced, so it will phenocopy what the AD2 mutant phenotype is going to look like, and it will give us a sectoring colony, and here I made the sectors in yellow chalk. Okay, so in this problem, what we want to do is we want to construct a double mutant that is mutated for both RAP1 and Sir3. So we want, and we have the starting strains here. We have a Sir3 single mutant, and we have a RAP1 single mutant. We want to make the double mutant between those. And so remember, in yeast, or in almost all organisms, whatever is wild type, you don't usually write in the genotype. But here I've written the wild type versions of the genes in these particular strains so that we can more easily follow the genotypes and the phenotypes. And so we want to make a double mutant, so we need to dissect tetrads, and we need to find the right class of tetrad that's going to have the combination of mutations that we want. And so here I set up the chromosomes as I often do, with the two homologous chromosomes here, the mom and the dad, and the other set of chromosomes over here. Now I set up the chromosomes in such a way that I have the genotypes lined up the way I want them ultimately to form this double mutant. But remember, and this becomes a non-parental ditype because it doesn't look like either of the parents. And so remember, we get equal numbers of parental ditypes and non-parental ditypes because of Mendel's law of independent assortment. We could just as easily have had the RAP1 wild type and mutant chromosomes switched and segregate. Okay, but this particular tetrad that we're interested in is a non-parental ditype, which would have our Sir3 RAP1 double mutant. So what would that look like? Whereas on the problem, there were 4 colonies to show the phenotypes for, there's really only 2 classes, so here I'm just showing the 2. So for the wild type, we would expect that with our AD gene inserted into the silence region, we're going to get this variegated colony phenotype with the red sectors. And so this would be that same wild type phenotype. Now, if we didn't have Sir3 in wrap 1, we would not get this silencing here. And so we should end up with a colony where add2 is being expressed. And if add2 is being expressed, then these colonies should be white. There shouldn't be any silencing associated with them. And I do want to point out that there is an error on the first version of the key that I uploaded, but I think I've now corrected the error that was the present before. Okay, so this is part A of question 4, and next I'll make a video to cover part B.